Hello everyone, this is Sean Taylor, Field Application Scientist Manager for Bio-Rad in Canada, and I will now demonstrate how to use ImageLab software to perform the analysis of quantitative Western blot data. Of course, the first step in the analysis of a quantitative Western blot is to normalize our target protein of interest by some form of a loading control. In this case, the loading control we've chosen is the stain-free blot image of the transferred total proteins in each lane. And this is actually becoming a much more common methodology for normalization and a much more accepted methodology by the journals. So we have our stain-free blot image, which is the total transferred proteins on the membrane itself. And we have our target protein in a separate image after going through the blotting procedure and the washes and so on. This particular target protein of interest was revealed using a fluorescent secondary conjugate, but this could have also have been a chemiluminescent image from an HRP secondary conjugate as well. The stain-free technology works perfectly well with both types of secondary conjugates. So there are two methods to perform normalization. One would be simply exporting the densitometric data from the target protein to Excel, the densitometric data from the total protein to Excel, and then manually performing normalization. The software also has a tool for doing this. So the first step in the process in using ImageLab software is to open the two image files, the target protein and the normalization image. And again, this could be also a normalization image from a single protein if that were preferable to use. In this case, we have the image open for stain-free, which we're going to use to normalize. So we open the two images, and then we merge them into a multi-channel image. And that's by clicking on File, Create Multi-Channel Image. So then we can merge the image, the two images, by dragging them into two different channels to create a multi-channel image. Now, a multi-channel image can also be created on the imager itself, and that would generate a multi-channel image of the membrane and the target protein as well. Here I'm doing it with two separate images that were taken. Click OK, and then the multi-channel image will be created. I've already created a multi-channel image for these two images, so I'm going to just minimize them, and I'm going to maximize the multi-channel image. So I'm just going to maximize the image so we can see things a little better, and now we have the multi-channel image created from exactly the same two images we just saw. So we see stain-free blot and our fluorophore for our target protein. The next step in the process is to select lanes and bands. Typically this would be done on the stain-free blot image because the lanes can be more easily detected, and then the lane image, the lane template, can be copied from the stain-free blot to the target protein, which is what was done here. Then of course the bands would need to be selected, and this is all described in the how to perform densitometric analysis video that we recorded earlier. For the stain-free blot image, there are two ways to select how to normalize. We can either normalize by total protein, which would be all the proteins in the lane, or we can normalize by a subset of proteins in the lane, and that's actually my preference. So if I click on bands, so in the lanes and bands tool, and then after I've selected the lanes, of course, I would select bands, and then I would add individual bands, which I did here, one band per lane. Once a single band in each lane has been added, and it can be quite arbitrary where you add these bands, then click the Adjust tool. And that permits access to these dotted lines on either side of the band that can be moved as I'm doing here. So we can stretch this region to encompass a particular region of bands around each lane. Of course, the same region should be selected, and everything between the two boundaries that we're setting here by these dotted lines will be used to normalize. So the software will take the sum of the density of all the bands in between these markers. So of course it's very important to set the markers at the same place on each lane so that we're being consistent with the normalization of the proteins that we're going to use to normalize by. I usually select a region of the membrane where I have good signal to noise and no artifacts of transfer like air bubbles or regions of large amounts of density that could be at the bottom or at the top of a lane. So this permits me to be a little bit more precise in the normalization using a more defined region of the membrane. Of course, I would have selected the bands for my target protein of interest for the opposing same lanes. So this is lane 1, 2, 3 and lane 1, 2, 3 for the stain-free blot. Very important to also just be sure that when you select, particularly on the stain-free blot, the lanes that you wish to normalize by, that you use the lane profile tool, and this is again described in the how to perform densitometric analysis video. I'll just review it quickly. But use the lane profile tool to assess that the appropriate peaks were selected, which correspond to the bands, and the software will take the area under these peaks of density for normalization. These blue lines are the same outlying markers that we chose, which define the region of each lane that will be used for densitometric analysis. What's really important and why I'm showing this is the background subtraction. We want to be sure that this blue line we see here is joining the lowest valleys around these peaks. And that can be done by setting the slider bar here. So this would be not enough background subtracted, and this would be too much background subtracted. The key is to subtract the background so it's touching the lowest valley around the bands. So this is around 18.4. I believe I had it set to 24. You can set it to a particular number. If I set it to 20, for example, I can apply that background subtraction setting to all the lanes. And then just click through the lanes here to just be sure that it's consistent, which it is. Of course, assess also the target protein to be sure that that also has been selected properly using these tools to select the peak, which is the bandwidth, properly, and the background subtraction, and click through the lanes to be sure that everything's been selected properly. So now I've selected the region that I want to normalize by my stain-free blot, the total proteins, and my target protein of interest properly. So now I'm ready to actually tell the software to normalize. So that's by clicking again in the Lanes and Bands tool under Lanes. And down here I have a normalization channel. So I can tell it I want to use the stain-free blot to normalize. So this would be which of the images contain the normalization data. In this case it's the stain-free blot. And you can tell it what lane to use as the reference lane for normalization. I'll describe this when we look at the data. I'm going to choose lane 1. And this is very important. So I can either tell the software to ignore the settings that I manually set as outlying markers for the region of each lane that I want to quantify for normalization, by choosing total lane protein. And then what the software will do is it will choose all the bands, background subtracted, in each lane. If I choose housekeeping protein band, then it chooses the specific region that I chose in each lane for normalization. So now I'm ready to, so I'm choosing housekeeping protein band, so I'm choosing the specific region, but I'll go back and try total lane protein in a minute. So then I can click on the analysis table, and the analysis table now shows me the normalized data. So the stain-free blot, which was the image that I used to normalize by, I chose lane 1 as my reference lane, as we see here. So what the software will do then is it will choose lane 1, the adjusted volume, which is the normalized, which is the background subtracted density of the region that I chose for all of the proteins that are within that region. And then it uses this as the normalized reference lane. So we're dividing the rest of the lanes that I chose for normalization by this lane to create a normalization factor. So of course it's 1 in this lane because we're dividing this value by itself, the adjusted volume. But in lane 2, the normalization factor is 1.3 because we're dividing 262, which is the density, the adjusted volume of the region that I chose, 262, and we can tell it's actually kind of decreasing a little bit, the density from this lane to this lane, so it makes sense that it would go down. 342 divided by 262 is 1.304. In the next lane, we can again see the density is decreasing a little bit, so it drops to 176. 342, which is the reference lane, divided by 176 is 1.939. So this creates a normalization factor to the reference lane, and then all we're doing is multiplying that value by our data in our normalization lane. So of course the values should be identical on each lane because we're using the factor from lane 1. So 1 times 342 is 342, 1.3 times 262 is 342, and 1.9 times 176 is 342. Of course, these values should be identical because they're based on the ratios of this value to the opposing values. We do the same thing for the band of interest. So this is our target protein. So now we're going to use the stain-free image to normalize. So we multiply the adjusted volume of the target protein in lane 1, which is 64 here, 64 million. And we multiply that by the normalization factor we achieved from the stain-free blot. So 64 times 1 is 64. In the next lane, of course, the band is a little less dense, so it's down to 49 million, or 40, almost 50 million, times, again, the normalization factor is 64. And then the next lane, which has an even lower density, down to about 34 million for the adjusted volume in that lane, that's background subtracted volume, times the normalization factor we obtained from the stain-free blot, is again around 67 million. So we can see that these values for our protein of interest after normalization are very close, which makes sense because both the total protein went down consistently, as did our protein of interest. So there was no net effect of treatment, if we will, in these lanes. The effect that we see in this density decreasing is simply the effect of differential protein loading, which we've now normalized. So this result can be exported to Excel by clicking on this button here. It will open an Excel file, and we have all our data, including the normalized values here for the stain-free membrane, which should be the same, and of course our blot after we've revealed with our protein of interest. So this is the normalized protein of interest data. And we can continue on with our analysis from there by looking at fold differences. We could have chosen, instead of housekeeping protein bands, which as I mentioned is the region of interest, we could have chosen total protein. And if we choose total protein, The data change a little bit over here, so we can see that we're no longer exactly at the ratios that we chose before. Because with total protein, software is using the entire lane to perform normalization, the background subtraction from the whole lane and not just the region we chose. And there could be differences in a total lane, particularly when we have very low signal to noise at the bottom of a lane and near the top of a lane. And you have to be very careful about artifacts. So don't use total protein if you see large density artifacts within the lane that were consequent to transfer artifacts or maybe the dye front that was fluorescing, or large protein aggregates that are at the top that you don't want to include, because those large regions of density could skew the data completely and give you very similar density values between the lanes, even though they may be different. Same thing where we see less signal at the bottom or top of a lane. There's a lot more noise, a much lower signal to noise in these regions, which can modify the data a little bit when we're looking at normalization. And that's why I prefer typically to choose a region of each lane where I get the best transfer and the best signal to noise for normalization. But just to make sure that we understand, the calculations are done identically. So it's using, for total protein lane, the adjusted volume for the total protein, and it's creating a normalization factor. Unfortunately, the values between total protein and housekeeping protein, as we see, the adjusted volume on the stain-free membrane doesn't change. But in effect, it is changing these results here. So if you focus in on these normalization factors, when I click on total protein now, they do change slightly. And that's because it's using now the total protein, even though it's not registering it here in the adjusted volume. You can prove that to yourself simply by going back to bands, clicking adjust, and selecting with these tools the total lane instead of the partial lane, as we did before. And then if we choose the total lane, so now we're at 1.312 and 1.893. If I go back to lanes, that's for total. If I click on housekeeping, which will force the software to use those delineators that I just modified, then the values are almost identical because I've selected the total lane. So of course, we see that the values now are quite consistent here. If I choose total protein, they're a little less consistent, but still quite close. And then again, it multiplies the normalization factor I obtained from stain-free for my protein of interest. Again, be cautious about using total protein if you have artifacts, bubbles, or artifacts at the top or bottom of the gel that grossly modify the density of the total lane, and use a partial lane density as I showed before. Now, the other option to use when we work with ImageLab software is simply to export the data manually. So what we can do, I've again selected my bands in each lane. If I go back to the stain-free blot and click on bands and adjust, I've selected just the region that I want to use for normalization in the first 3 lanes, and I'm going to normalize in these lanes for the target protein. So if I click on my target protein image, I can then click on the analysis table. So I have the adjusted volumes of those target proteins, and I can export that to Excel. So that's in Excel now, and if I open Excel, I have that image. I'm just going to delete the previous one. So I have just that image for my target protein. Go back into ImageLab software. If I click on the opposing image for stain-free, the analysis table populates the data from that image. So that's the adjusted volume here for these lanes for the region that I selected. Then I can export that to Excel. So now I have another Excel spreadsheet open which is here for my stain-free blot, and then I can select those data, copy, and paste them in the opposing Excel spreadsheet for my target protein of interest. And then it's just simple formulas where I can say, okay, I want to normalize, so I will normalize my protein of interest, which is from the dye by the stain-free blot. So I will divide, like we normally do, the adjusted volume. So let me just stretch this a little bit so we can see. The adjusted volume, which is the background subtracted volume, this is the data we want to use, so equals from lane 1 divided by the stain-free blot, also from lane 1. And I can copy those results between the 3 lanes. So now I have my normalized data. We can see the normalized values are very, very close, as expected, because there was no effective treatment. The reason why our bands decreased in density from lane 1 to 3 was simply because we were loading less and less protein. It wasn't because there was an effective treatment. If you want to do the full difference between those lanes, typically we would divide the rest of the lanes by a reference lane. So we'll assume lane 1 is our reference lane. So we'll divide our reference lane by itself. Put a couple of dollar signs to make sure I divide the other lanes by the same reference lane. So that's 1, and copy that down. So all the other lanes are the same as the reference lane pretty much. They're all around 1 because, again, the density, the normalized density did not change between the treatments.